In this first unit, I will try to explain what a genome-wide association studies is, and it's important to understand the basis for quantitative of complex traits characterization and its relevance for future breeding in organic and low input cultivation. And I'm going to be based not only in giving the information, but I'm trying also to give some practical examples on how to do this for the people to practice in their homes. So maybe during the practical exercise, there's usually some doubts, or there's no problem if anyone wants to do the question.
Checking if everybody is following. Okay,
Exactly.
good. That for this case is okay, good.
So if as said, if you have any doubts during the practical lessons, or write in the chat or raise your hands, and maybe I can stop for a few seconds. Okay, so let's get started. The causal relationships between phenotypic differences and genetic variations in a genotype, which means the relationship between a gene and the phenotype and the trait, is a fundamental biological interest. So knowing the causing locus or loci, it's what we call the genes for a trait, is key for these breeding purposes. We have two main goals to know this relationship. Is first, knowing their genetic regions allow you to develop markers, genetic markers, to select genotypes with one phenotype of interest, like easier. For example, if we have a marker for extended shelf life, by applying the marker to a collection of varieties, we can select the best varieties for cultivation, like easier than with other techniques. Second, establishing correlations between traits and finding the genetic relationship between these traits genome might help to select one complex trait through another simple trait. For example, let's say that yield and, for example, length of the panicle in rice are correlated. So if we know that some type of panicle length is related to a better yield, we can select this panicle instead of the yield, which is a more complex trait. Okay, I see one person is not seeing the presentation. Is anybody else with this problem? Okay. Okay. Then if it's just one problem, okay, then I continue. Thank you very much. So let's continue. When we talk about qualitative and quantitative traits, we are talking about traits that are controlled only by one or few genes, or traits that are controlled by many genes. That means, please silence your microphones, because if not, I cannot concentrate properly. Thank you. So qualitative traits normally are follow the Mendel laws that indicates dominance of one trait in the first intercross and segregation in the F2 generation. And these traits are, let's say, quite easy to analyze, because you only need to analyze a cross of two parental lines differential for this genotype and the phenotype, finding easily the region differential between them, developing markers for this single region, and testing easily a collection for this gene, and thus the analyzing the collection easily for this phenotype. The problem is that most of these interesting breeding traits, agronomic interesting traits like yield, for example, or fruit quality, or tolerance to difficult agroclimatic conditions, or adaptation to different soils, are normally complex traits. That means that they depend on a lot of genes along the genome. So they are very dependent on the environmental conditions for the expression of this phenotype. So what we see is that we cannot categorize these traits into easy 2 or 3 categories, but they are normally a big, we have a big interval, let's say, of phenotypes. For example, in the number of locus that you can see here in the screen, we can have 2, 3, 4, 5, up to 8 locus, which is not like established categories. It's the same that happens in height, for example, in the plant height, that we cannot say there's categories, but there's a normal distribution of this trait in a population. So analyzing these traits, both phenotypically and genotypically, it's very complex because they depend on a lot of genes and also on the environmental conditions. For the last 2 decades, Genome-Wide Association Studies, or GWAS, have been a successful approach for associating numerous polymorphism in the genome to complex traits. Let's say that we have been able to associate a complex trait like yield with different markers that would be the different genes that depends on in the genome. So in this case, we normally do not talk about genes, but about quantitative trait loci, or QTLs. These QTLs would be each one of the genes or the genome or the regions in the genome that are acting for the occurrence of one phenotype. That means the accumulative effect of all these QTLs is the phenotype that we can observe. These studies, these GWAS studies, are very interesting because they exploit the existing natural variability within a population generated through the years after selection and recombination through the years. So it can be performed either on a collection of varieties, traditional varieties, or a collection of breeding varieties, or different types of breeding populations that needs to embed all this diversity for the trait of interest. As explained in previous modules of this training, traditional varieties and land races are considered now a valuable resource because of this variability and are very useful in this type of experiments. So they show a high added value for this type of experiments, because in one single population, a collection of these traditional varieties or land races have a lot of variability phenotypically, but also genetically. So, okay. Basically, the GWAS would be based on phenotyping a population that needs to be diverse for the trait that we want to study, and then genotyping this population with different methods to obtain the genetic information, and we need to gather this information together to try to find if there are regions in the genome that are differential between varieties with different phenotypes. Let's say that when we have different phenotypes, we need to find the regions that are making these phenotypes different within the population. So, okay, this approach presents some advantages, like it is not necessary to invest time and resources to create a breeding population because we can exploit the natural variability in, for example, a collection of land races. Also, it allows to analyze a wider range of diversity because it's based on recombination events in a population that can be carried out during centuries. It is useful for any trait we can imagine. We can work on several traits at the same time if the population is variable for these several traits we want to analyze. And we can obtain high precision in the gene mapping for genomic studies. And we can also associate complex traits like adaptation to different agroclimatic conditions, low input conditions, adaptation to organic breeding conditions, and like very complex traits, tolerance to stresses, and that's it. That cannot, that have been impossible to associate in other with other technologies. It is also true that these studies present some disadvantages. For example, we need to be sure that we have availability of diversity for the traits we want to study in the population, not only phenotypically but also genetically. So we need to be sure that even two phenotypes are different, it also relates to two differential genotypes. As I said, the population must show variability for the trait, not only phenotypically, but also genetically. And we need to avoid some problems with the population. I explain myself. Population stratification can cause bias or fake associations between trait and the genome. Okay, I'm going to put some examples. For example, to avoid a fake association between, spurious association between a trait and the genome and the genes, we need to be aware of the population structure. For example, if we have fixed alleles in the population, we suppose that in the study, in the GWAS study, we are supposed, as I said, to represent all the possible variability phenotypically and genetically. We represent all the alleles of the genes that are, let's say, controlling the trait we want to study. But if we only have two combinations of alleles, which that happens in this case, that we only have A and B fixed, A majus, B minus, and A minus, B major, so we might associate one of these alleles to the phenotype when it's not really the association. This would be an spurious association. We can associate these two genes to the phenotype because they are differential in different phenotypes, but they are actually not causing the phenotype just because these alleles are fixed in the population, because other combinations are not appearing. Maybe if all the combinations were appearing, we would see that it's not actually related to the trait. Another example would be ligated genes, or what we call the linkage disequilibrium. Basically, two proximal genes are always inherited together, and as they are very close and inherited together, they would be both associated to the trait, even if one of them it's actually causing the phenotype. That means we cannot distinguish them. So it would be causing a fake correlations between one gene and the trait. This can be overcome now with sequence-based genotyping strategies that give a lot of information about the genetic haplotypes, the combination of genes and alleles that are present in the population. So in summary, we need to have in mind the existence of genetically similar individuals that can add some bias, some problems to our analysis, and can add some fake associations. So numerous examples have been performed in a lot of probes and are available in repositories and are available in Google Scholar and and published in a lot of sources. So, for example, in eggplant, it has been discovered this last year some genes that are associated with the pigmentation of the peel and thus the quality of the fruit, and are very interesting for further studies about this fruit quality for eggplant commercialization and cultivation. And also, for example, in tomato, 2 or 3 years ago, some genes or some QTLs were associated to locule number, which is also very important for fruit quality in tomato and also commercialization of this tomato, because as you know, as you might know, land races tend to have more locules, and commercial varieties tend to have less locules, and less locules tend to be more commercial. But it's interesting to have everything of this in mind. So now I am going to show an example on how to perform a GWAS analysis with the software Tassel5 in 2 different trays, and we are going to comment and compare the differences. So Tassel5 is a free software. You can find the link to download it in the additional resources of the presentation. Here in the additional resources, you have here Tassel5, where you can download it and how you can install it in your computers. This would be important because your homework would be to try to perform your own analysis in this Tassel5 and send us the results. So everything you need for doing this practical exercise, as well as your homework, is ready in this link for GitHub. Everything is there: the presentation, the data you need, the data I am going to use, everything. So if you have any problem, you can go there and is everything there. Also in the presentation, I prepared all the screenshots of the analysis I'm going to perform. So I'm going to perform it now on live, but you can follow also the presentation, and you can do your homework here. Okay, you have time. You can follow your close your mic, please. You can follow my presentation here, and also you can do your homework with the presentations. Okay, as now I want you to follow me, I am going to explain it first, and then I send the link to the chat, and you can do your practical exercise. I leave you time to do the practice. Okay, and as you see, you have all the screenshots of all the process, so that you can follow me and you can try later your own practical exercises, but I am going to do it now. Okay, let's start. So we need now to see the differences between a whole genome sequences and a genome by sequencing. We have different technologies to obtain genetic information. The first, as we all know, is the whole gene sequencing of a whole genome in any crop. But there are other technologies to obtain genetic or genomic information. For example, genotyping by sequencing only sequences one SNP, one base, each X number of bases. So they return you a very long list of bases, which are not sequential in the genome, but separated in a X number of bases. But this technology also returns a lot of information. So with GWAS, we have enough information to know genetic differences between within a population, let's say. So the first practical example would be a GWAS analysis in a collection of tomato, Solanum enrico-persicum, a core collection with accessions of very variable tomato land races. And they have been phenotyped. All the core collection of tomato land races have been phenotyped for growth habit. And we have a phenotype document with 3 classifications: determinate, semi-determinate, or indeterminate. Okay, so as we talked, for the GWAS, we need phenotypical information and also genetical information. So let's start with tassel. Okay, I'm going to, you see the tassel screen? Okay. So we're going to start uploading to tassel the information of the phenotype. All the, let's see, open as, we're going to select phenotype. Okay, I'm going to select grow habit txt. And as you can see, here we have information of 353 accessions and the beta or the average for each accession. Because as you know, to do this type of experiments, we need several data for each accession. This would be the average, the mean for this accession. Okay, now that we have the phenotypic information, we're going to open the genetic information. Yes, make Vegas, keep that, okay. And we are analyzing growth habit. It might take a while depending on how fast is your computer, but here we have all the information, all the genetic data we have for each accession. And you can see here all the genetic bases in the genome, but the positions that would be this in the upper side are not correlative. That means that we have one base each an X number of bases. So now that we have this data, we're going to see which is its distribution in the population. In the let's say the population is our collection, our study collection, our study population. We are going to see how these individuals are related because of what we said previously. If this population has some issues, some relationships within the individuals, we can have some bias or some fake associations trait gene. So we're going to do a PCA, which is an easy way to see if they are correlated. Okay, now we're going to do a chart representing these values. Okay, I don't know if you are familiar with this type of graphs. This is a PC1. What we have here represented are the individuals of this collection, and we can see that there are more or less individuals all over the graph, but there's a higher amount on the right part of the graph. That would mean that some individuals are more alike, are more related than others. The program now needs to have in mind this type of relationships between the individuals to do a proper analysis GWAS later.
Neus,
So,
sorry. On the
yes.
screen, we see the table and not the PCA graph.
Okay. Okay, there's no problem. It's in my presentation in PowerPoint. Sorry, I don't know what it's not displayed on the screen. But well, it's not problem. What I wanted to say is that we need to take in account that individuals might have some relationship among them, and we need to to tell the program that this needs to be taken in account to avoid bias during the GWAS analysis. So what we are going to do to tell the program that these relationships can occur is do a distance matrix with calculate the distances of each individuals with the rest of the individuals, and then an MDS. In this case, we are just telling the program, have in mind that these individuals have this relationship, and this can cause bias. So avoid the bias caused by these relations. Now, easily, now that we have everything in the program, we need to intersect the information of the sequence, the information of the phenotype, and the information of the MDS, that would be the correction of this bias caused by the relations among individuals, and we are going to intersect all this information, creating one unique matrix. Okay. So now it's easy because the program does everything. We just go to association and we do a GLM analysis. It's a general linear model, which is a simple statistical model to do GWAS, but taking in account these relations between individuals. So this can take a while. Okay, let's see. Okay. Okay, easy. And now we're going to see the results. To see the results of GWAS analysis, we have two main, let's say, two main graphs. One would be the cuckoo plot. I don't know if you are able to see the cuckoo plot. Okay, the cuckoo plot would be all the individuals, how they are distributed, respect to what was expected.
Neus,
Okay.
sorry, we cannot see on the screen the cuckoo plot.
Okay, then here. So we have done this PCA. Okay, this would be the PCA resulting from the first analysis. Then we calculated the distance matrix, we calculated the MDS matrix, we combine all the information together, the phenotype, the genotype, and the matrix of distance. We did the association analysis with the statistical method GLM, that is simply the most easier or statistically simple one, but it has into account the distance matrix. And this would be the cuckoo plot. Okay, basically what you can see here is the distribution of the markers, okay, the SNPs, the bases that we have seen according to the expected. The expected would be the hypothesis that any marker is related to the trait, but as we can see here, the markers are going up in the distribution and are not following the expected. So that would mean that some QTLs are actually related to the trait. And the other representation of this association between trait and genome would be the Manhattan plot. That this is just, as you can see here, is just each base pair sequenced, and after some filtering, each base pair represented along the genome. You can see the positions in the genome. And if the QTLs go up some level, which would be the blue line, all the QTLs that go over this line would be statistically associated with the trait. After we know which QTLs are associated with a trait statistically, we need to select the most significant ones. And the QTL, let's remember that it's only a base pair, a genetic base in the genome. We select the most significant ones and we establish a region upstream and downstream, because we don't have all the bases sequenced, but only one sequence in an interval. So we select a region upstream and downstream, and we look for information in this region, and we use available genomic information in databases to check if there are any annotated genes in this region. Okay, so we analyze the function of the annotated genes in this region, and we analyze if they could be involved in the expression of the trait, if they have agronomical sense or not. And if we don't find this type of information, we can search also in information of other very related species and other databases. For example, if I have a region and I have the sequences and I have the genes for eggplant, for example, but I don't have much information, we can have for these sequences, these genes, in other databases in related species, like other Solanaceae, more studied, like tomato. Okay, I understand that for the ones that have not seen this type of analysis before, is something complicated. So now we have another example, which is a GWAS analysis in a collection of Solanum persicum using another trait. It's the same collection with less individuals. And I used another trait. We phenotype this smaller collection for fruit shape. You have here all the fruit shapes we used. And you can
Neus,
do your own example. Yeah.
Neus, sorry. Okay, maybe for this example, okay, you can offer the opportunity the people asking questions, okay, because this is true that in 15 minutes showing a program for some people who is not familiar to them, better to ask questions directly. Okay, I think it's very important.
Yeah, what I wanted to say is that for me, it's not either easy to be presenting and being on the chat all together. So
Yeah,
now
okay.
that we have some time, you can, I can show the example, but you can also be following me, because I'm going to send you the link to my GitHub page. Okay, let the chat. I'm going to send you the link to my GitHub page. So here you will find the presentation, the phenotypic and genotypic data I just use for the growth habit example, and also the information for the fruit shape genotypically, and all the information genetic and phenotypic for fruit shape and everything. So if you want, I give you 5 minutes to download the program and everything. I share you here. Where can you download Tassel? I give you 5-10 minutes to download Tassel. Okay. So you can follow me, and then
Good.
We will do it together. And you can start maybe later. I give you some time to solve some doubts and everything. I understand that you cannot know or learn how to use a program in 10 minutes. This is not the idea of this course. So my idea is not for you to learn how to use Tassel. This is not my idea, because there are a lot of softwares you can use and a lot of tools you can use to perform a GWAS. So my idea is that you do not memorize how to use Tassel, but my idea is that you understand what are the steps we need to do to perform a GWAS analysis properly, and which is actually the importance of this tool.
Okay, good explanation, Neus. But maybe the guided exercise now will be better for them to understand, because there is people familiar to this kind of programs, etc. But maybe there are other people who are not so familiar or used to work with them. So maybe, yeah. But I think that the practical exercise, a bit guided and following you,
Yeah,
will
sure.
be much better. Okay, thank you.
Sure, but as just in case anybody just loses at some point, there's no problem, because in the presentation you have all the screenshots of all the steps I am doing, as you saw. There's all the steps, so what, even you are lost at some point, you can go to the presentation to do your homework at home. You will have plenty of time. You can just follow all the slides in the presentation, and you will go to the result with no problem. Even if you are lost at some point, following the screenshots in the slides of the presentation, you will be fully able to get your results with the homework. Do not worry, it's all prepared for you to follow this unit. So I give you 5 minutes more, and I start with the second practical example. I think the most important thing we need to retain from this unit is that knowing the genetic basis of the traits, moreover of complex traits, quantitative traits, is very important, because most of the agronomic traits we want to improve during breeding processes are very complex. Yield, adaptation, tolerance, resistance, all these complex traits, adaptation to environmental conditions, to low input supplies, all these traits depends on a lot of genes. So knowing a basis of these of the control of these complex traits is a very important base to select the varieties that will work better in different conditions. So now I'm going to start. I will go slowly, okay? As I said, we have seen the practical example of growth habit in tomato. Now we are going to see the practical example of fruit shape. Tomato has a lot of different fruit shapes, from flattened to rounded to piriform, ellipsoid, a lot of variation. So we selected a collection which phenotypically was very diverse for this range of phenotypic diversity. So I'm going to open Tassel now. Here. Okay, let's see if in this way we can, you can see the graphs. I'm going to remove all the data from the previous analysis. Right. The data sets. Association, sequence. Now I have an empty screen. screen. So let's go to open the phenotypic data. So open as, file, open as, and we search for phenotype, okay? And we open our phenotype. In this case will be fruitshape.txt. Okay, and now, as in the case before, we have here all the accessions, okay, and it's average for these shapes. We have some accessions with type 1 of fruit shape, some of form 2, some with form 3, some with form 4. Let's say it's a variable population with a lot of differential fruit shapes and phenotypes. We can see here that in this case, we only have 186 collection. Okay, this is number of rows. That just means that we have 186 rows, 186 accessions. Okay. Now we're going to open the genomic data as make best guess with keep depth, which means that we are going to retain all the genetic information in the big file. Okay. So, okay. And we are going to open this fruit shape 186 fruit shape.wcf, which is the file format that we normally use for genotypic data, for genetic data. So we open this file. Okay, so as we saw before, we have here all the land races, all the collection, and here the position of the markers that have been sequenced. The marker is just one base, one SNP that has been sequenced. Okay, and here you have each result, each base for each position in each land race, in each accession of the collection. With this, the best part of this software is that it's very visual, so we can see what we are doing. Is very easy to understand, and we can follow it quite easy. As I said, these are not all the markers, all the positions that have been sequenced, but the filtered one, the best ones. We filtered for quality, we filtered for significance, we filter for a lot of things, but basically these are the best quality markers that we are going to use. And we have a total that you can see here of a total number of sites of positions of 4249. That it's actually a lot. So now we are going to see how is the distribution of our population by doing a PCA. So we go to analysis, relatedness, because we want to know how our individuals are related with each other, and we do simply a PCA. Easy. Just go to OK without touching anything, and we have here the calculations of the PCA. Now to see these results, we go to results, to the chart, okay, and we change from histogram to XY plot, and in X and Y we put PC1 and PC2, which are the main components of a PCA. So here we have the distribution of the individuals, of the 186 individuals that we have in this collection. So what we can see here is that even there's a high distribution of these individuals, this cluster of individuals at the right might cause some problems because they can be genetically related or related in any model, so that these relationships between these individuals can cause some bias or some fake associations, trait, gene. That means that maybe the program return to us some QTLs that are not actually related to the trait, but they have been related just because some relations between the individuals. So to avoid the program to tell us fake associations, what we do is tell them, okay, calculate which are the actual relationships between individuals. So for doing this, we do analysis, relatedness, and we say the program, okay, calculate which is the actual distance, genetical distance of these individuals. Wait. Okay, we need to select the sequence, the sequence database, okay, to do this analysis. Okay, so we select the sequence database, we go to relatedness, and we say, let's do a distance matrix. And easy, we have here all the relationships, all the calculated genetic distances between each individual with the rest of the individuals. Okay, and now we're going to say, let's take this, let's have this distance matrix, these distances into account to do the GWAS analysis. So we do an MDS. It's like a multi-dimensional distance matrix. I'm not going to, it's like a PCA, but taking in account the genetic information about distance, genetic distance among the individuals. Not going to bore you with this. So now we have this MDS PCA matrix. We are going to select this information about the genetic distance between the individuals, the sequence. Okay, we select the three at the same time with control button, and the matrix, which is, and the numerical, which is the TXT. So we select fruit shape, MDS, and the sequence. Okay, and we go to analysis, association, GLM. Okay, I am thinking on so many things that I have not my head. So we select the three databases and we go to combine. We're going to combine them with intersect join. So we intersect all the information, which means that we have in the same database the genotype, which is here, the phenotype, and the PCAs, which are just telling us which are the genetic distances between individuals. Okay, so we select this database combined, where we combine all the information, we go to analysis, and now we do the GLM. Just say okay. Okay, and the program just performed the GWAS with the GLM statistical analysis, which is a simple one, but that has into account the distance matrix. So I suppose some of you are writing into the chat. I'm just finishing showing the cuckoo plot and the Manhattan plot, and I go to the chat to solve your any doubts, okay? So after we have combined all these and we have performed the GLM analysis, we go to the results and we can see the cuckoo plot, just okay, which would be this form. If the hypothesis for the expected values, which is the black line, the expected values would be the distribution of the markers, of the dots, of all the positions, if any QTL were associated with the trade. So if any QTL would be associated with the trade, this line, the average of these points, which are just the markers, would be on the black line. If the QTLs are associated, if some QTL is associated with the trade, what we will see is that the points are like going on the expected values, like far from the expected values. In this case, we can see that more or them, the tendency of the points of the dots is following the expected values. There are no points that are very far away from the expected values. That could mean that any QTL is actually or statistically related to the trade. Let's see now the Manhattan plots to see if some QTL is associated. So, okay, normally we calculate a base, a value from which we consider a QTL statistical associated with the trade, which is around 6. So here we have the log 10 value, and we can see that no QTL is surpassing this 6 value of the logarithm that I say is more or less the threshold. So in this case, with the data we have, with the phenotypic and genotypic data we have, we are not able to relate this trait with any QTL, with any genomic region, okay, in the genome. That doesn't mean that there are no genes controlling this trait. Of course, there are genes that control the expression of this phenotype. But with the data that we have, we are not able to see, to analyze or associate these genes with the traits. Okay, I'm going to see the chat. Is it possible to assure we can see? Okay, now I'm going to share the presentation because I have the results there, and we comment the results, okay?
Neus, Neus, there's a question from Ricardo Fratti. Okay.
I didn't understand how you set the threshold in the Manhattan plot. It's just a statistical calculation. It's just a settled statistical calculation which puts the alpha in the 0.05, which is normally is the statistical percentage.
Neus, could you please use your slide where you are managing this? Also explain that. Okay. It's much better. Did you explain the question with the graphics?
Yeah. I didn't want to enter a lot into the statistics because I really think that it's very complicated to understand in a to our presentation, or maybe in a 10 minutes practical example. So I'm not actually explaining how the statistic is made, because it's something complicated. The methods are not easy, and I think it's more with it's more related with the biotechnological and the programming statistical methods. So what I'm going to say is, we normally do the analysis so that we ensure that the 90% of the results are okay, and only a 5% of the results could be false. So a 0.05 of the results could be false with our analysis. So I think somebody's controlling my presentation now. So with taking in account this 0.05 of false results, we calculated the logarithm to ensure that these markers that are over this threshold are actually related to the trait and are not a false positive. Okay, let's say that this threshold says to you that anything above is actually related to the trait and are not a false positive. We are confident of this result. I know what the p-value is. So the calculations are based in the p-value, but I'm not going to show you how to calculate this base, because I do not think it's interesting at this point.
Yes, Neus, as Neus is telling, is just a statistical threshold. Okay.
Exactly.
Is that over the 6 value, then you can trust that there is something real in this or this or this, in this part of this chromosome related to the expression of the trait. Okay. Maybe it's a bit complicated to explain how this statistical proof is works. Okay, but it's just a question of statistical trust. Okay, over the 6, any point, any bubble over the 6 value means that okay, there are several points, which each point represents a location in this part of the chromosome. Each color represents a chromosome. Okay, is related to the trait, to the expression of the trait.
Right.
Higher or lower expression of the trait, but it's related. Below, it's close, but there is not a statistical trust to trust it. Okay, it's just this. Okay, don't worry about the rest.
Exactly.
There is also another question about the Manhattan plot.
Yes.
From Ricardo Fratti and Noemi Peter. I don't know if Neus, you have answered properly with your comment.
How can you say a conclusion? Okay, there. Yeah, I think for some people this type of analysis is new, so I understand it's complicated to understand it in 1 hour. Or in 15 minutes. So as some of you are questioning how the Manhattan plot works, I'm just going to focus on the results, because let's remember, the important thing is that you do not memorize how to use Tassel, because there are a lot of tools or a lot of softwares you can use, but the important thing is that you understand which are the results. So let's remember, we open a phenotypic file with phenotypic information about a collection of plant races. Okay, we open a GWAS data file, and a GWAS is just telling us that we have some land races or some accessions, let's say, varieties, whatever, individuals, and we know which base pair in some locations. So in the same location, one variety has a T, one variety has a G, one variety has a C, whatever. We have all this information. Which base pair each variety has in each position. It's not all the base pair in all the genome. It's just some base pairs along all the genome. Okay? So you see here that you also have green and blue spots. That this is not a base pair. This is telling you that maybe this accession is not homozygous, is heterozygous. So it has both, has two, for example, has the two type of alleles for this position. But I'm not going, as I said, I'm not going to go further into this because it's very complicated. It's just what I want you to know is to analyze the results more than using the information. I think when some of you are, what is the extraction of the genotypic file? Basically, the genotypes, the position, and each one of the results of the bases pair sequenced for each region or each position, sorry. Thank you. Okay, so into the results. After we open both files, we do the PCA. We analyze to see how the population is distributed. We calculate the distance matrix and the corrections for these relationships between individuals. We intersect, we combine all the information, the phenotypic information, the genotypic information, and the relationships, the corrections we need to add to this analysis. Then we do the association analysis, and we see the cuckoo plot and the Manhattan plot. What we would expect in the cuckoo plot, if some QTLs were associated with a trait, is that the QTLs goes far from the expected values. Right? The expected values are the expected values if any QTL is associated with a trait. So in this case, in this last case, we saw actually the tendency of the individuals is the markers, sorry, is to follow the expected values. So as we see in the Manhattan plot, there's no QTL going above the line, the threshold we marked. So basically, a Manhattan plot is a representation of all the positions we have, okay, all the markers along all the genome. You can see from LG1, that would be the first chromosome, from the 2, 3, 4, 5 chromosome, 6 chromosome, each color of dots is one chromosome. So you have here the distribution of the markers of the sequenced positions along the genome, okay? And the logarithm value would be, let's say, the association of this marker to the trait we are analyzing. So as any of these, in this case, any of these QTLs are going above the line, we cannot say that with this population and this genetic information, we cannot find an association. Okay? How to interpret the cuckoo plot doesn't show possible association. If you see the cuckoo plot is a more or less a linear distribution of the markers, and any of the markers is surpassing like far the line. If you see the other example, see how far the dots are from the expected values. We have to take into account that anything is perfect So the tendance of these dots is linear. The tendance in these dots is linear and close to the expected values. While if we look at the other example where we have QTLs associated with the traits, we see that the traits are quite far from the expected value. Okay. So now I don't know if you have had time to follow these examples. As you have all the information for doing both practical examples, I leave you now, let's say 15 minutes, okay, to do both examples, okay, and we comment. And if you have any doubt using Tassel, now is the moment to ask, okay, so that you can do homework later. I leave you 10 minutes to do these practical examples, and then we continue, okay?
Neus, just to finish, that there's also another question from Neus to GWAS. How to interpret? Okay.
Yeah, I answer it.
Okay. Have a look to the chat and answer, please.
Yeah, I leave you 10 minutes, okay? I leave you the fast quiz. So when you are resolving the first and the second practical exercises, if you want, you can answer this fast quiz and we can discuss, okay? Okay, so I see some of you have already answered the fast quiz. I just want to, I don't know if I would say start a debate. I just want to comment two things that we need to take into account to this type of analysis. I am being aware. I am sorry for the ones of you that are very good at the statistics, but my idea for this unit was not to get very deep into the statistical analysis or even type of statistical analysis we can perform. But my idea was to give a hint on why these type of analyses are important for breeding, and why understanding the genetic basis of our trait is important for doing breeding, for doing breeding more efficient, for doing breeding more understandable for all and everything. So going to the questions. So how can the results be affected by the existence of two subpopulations? As some of you commented, we suppose that in the population of study, we only have one population that represents all the possible phenotypes, more or less equally, and we represent also all the combination of genes and all the combination of alleles of these genes that will lead to the phenotype. But if we have two subpopulations within the bigger population of study, what we could have are more representation of one allele than another, even in appearance of one allele or one combination of genes that can cause false association gene trait, or even that we do not obtain some association of this gene with the trait just because we don't have the combination of genes and alleles we need. Okay. Exactly, because it is possible that it can appear fake results. Exactly, it's possible that we have linkage disequilibrium, ligated genes. We can have some structured population with some haplotypes and not other haplotypes, just combination of alleles, and that's it. Okay, I just wanted to remember you the importance of having all the variability represented in your study population. Okay, it's because it can happen can happen in this case that we do not have represented all the alleles in the population, and we are not able to make a real correlation between gene and allele, or also another thing that would happen. Okay. And the other question, which could be the reason why in the case we do not obtain any associated QTL in the second case? As we saw in the first example, in the first practical example, we obtain some QTLs associated statistically with the population of study, or with the trait, sorry. So we obtain some regions that have genes, okay, that are interfering in the expression of the phenotype. But in the second case, we didn't obtain any QTL, any genomic region, any gene associated with the trait. That doesn't mean that this phenotype do not depend on gene. All the phenotypes depend on the expression of a gene. What that means is with the population that we have, with the information we provide, we are not able to find this relation gene trait. Okay, so basically what could be happening is we do not have enough variable positions in the genome. What I mean is we have not sequenced enough positions, or more probably, when we filtered after we receiving this with this genetic information, we have filtered for quality SNPs. Maybe we have filtered so much that we have lost a lot of markers that are actually important for making the genetic differences between the varieties. Also, what could be happening is that our population is too small, okay? So that we are not representing all the phenotypes and the genotypes. So maybe what is happening is that we are representing not enough individuals to be able to statistically to be sure that we can establish a relation with a short relation between a trait and a phenotype and a genotype and a gene. Okay, in the first example, we used more than 350 individuals. If you see here, we are using 353 individuals, 353 rows. I don't know here. Okay, here in the information, we have 353 rows, which are the individuals. While in the second example, we are only using 186 individuals. So that's another example why selecting a good population of study is very important to perform a GWAS analysis. Okay? So that's it for the fast quiz. Now, the ideas I want you to remember from this unit in this module is that, from this session, is that GWAS has been a technique that has been widely used in a lot of crops to associate traits with genomic regions, more complex traits which are dependent on a lot of genes and not only one gene. So, as I said, to do this, to do a GWAS, we need a non-structured population, a variable populations which have not only a lot of phenotypical variation for the trait that we want to study, but also genetical variability. Specialized programs like Tassel are required to combine this phenotypic and genotypic data and obtain a result and do a GWAS analysis. But we use Tassel, but there are a lot of other methods. Okay, you can find them online. And after we obtain these QTLs, these regions associated with the trait, we will need to analyze these regions separately to find candidate genes, which are actually responsible for the expression of this phenotype. Okay? So this is the proposed homework. The proposed homework is that you try to perform your own analysis with Tassel-5 and the data that you can find in additional resources in the GitHub link that I sent you. Okay, it's also here in the presentation. Okay, you have the Tassel, you have data, you have all the data in this link, in the GitHub link. Also, you have the presentation to follow the slides and go slide by slide, step by step, like slowly. And the proposed homework is that you take the VCF, which is the genome data that is called genotyping data VCF, and the phenotypic data, which is phenotypic data.txt. You take those two files and you try to perform with Tassel your own analysis. Okay, Tassel is available for Windows, but also for Mac. You can try in your computers. Okay, and you after you do that, you have sometimes to send to me and Petra and Adrian the results of your analysis. You can send me a screenshot of the cuckoo plot, also a screenshot of the Manhattan plot, and you need to explain to me your results. Okay, you need to explain to me if you get QTLs, if you can locate these QTLs in the genome, okay, because you have the groups of the chromosomes. Okay, you can say we find these QTLs in these chromosomes. Okay, and if you obtain less a little number of QTLs or you do not obtain them, why is it possible that you do not obtain any QTL? Okay, so that would be the proposed homework.
Just a quick correction.
Yeah.
They are supposed to send the homework to me, not to Petra. So I will write down my email into the chat. Okay. So just so you all know. So you send your homework to Neus. Her email is visible at the presentation, and I will write down my email into the chat.
However, I will change the presentation in the GitHub also, so that they have it correctly. Here. And that's it for my part. So thank you all of us for attending, and see you in the unit 4 of this module 4.